although some of the participants really love the music, but it's 10:03 and I think it's time to start. Welcome everybody. My name is Diana Ionescu. I'm a pathologist at the BC Cancer Agency in Vancouver, British Columbia, Canada. And I will be the moderator of the second presentation from Camppods. Good morning, good evening, good afternoon. We have almost 100 participants today again from multiple countries. It's always my pleasure to see how the online meetings really brings the world together. We have participants today from Australia, Bangladesh, Belgium, Canada, Qatar, Romania, Saudi Arabia, Singapore, Switzerland, UK, and United States. So I would like to welcome everybody. I think if people do see me, then probably they also see the window with the overall learning objectives. So just not to be late on the program, I will just go over the overall learning objectives for Camppods. When we established this webinar series, we really tried to, first of all, establish a strong Canadian oncology education network, which now it looks like it's becoming international, so maybe we'll have to change that first learning objective. We really are looking for a forum to compare ideas, thoughts, and common challenges, to identify gaps and find team solutions for improving patient care, and to integrate pathology and oncology practices. If I can have the next slide, please, Taylor. Camppods are accredited for 1 hour of CME through the Canadian Association of Pathology and the Royal College of Physicians and Surgeons of Canada. Next slide, please. I would like to thank the sponsors of this activity, AstraZeneca, Bayer, and Merck, for making this webinar possible. We're definitely looking forward for their support to continue Camppods beyond November. Next slide, please. Just a few housekeeping rules. Please make sure you unmute your microphone and also stop your video. And if you would like to chat, submit your questions in the chat or even chat privately with any of the participants. Just as a reminder, this session is recorded and the chat is part of the recording. So please be careful what you write in the chat. Next slide, please. The best way to view the presentation is if you go on the right upper corner of your screen and click view, and that's the best way to share the screen. During the presentation, we will have a number of polling questions, and you will be asked to submit your answers by selecting the correct answer and then pressing the red button that says submit. Next. So it is my pleasure to introduce today's speaker, Dr. Cheryl Matter. Dr. Matter is a pathologist at Alberta Precision Laboratories. She was trained in anatomical and clinical pathology and gastrointestinal and liver pathology at the University of Washington, followed by subspecialization in molecular pathology at the University of California, San Francisco. Currently, Dr. Matter is the medical lead for molecular pathology North in Edmonton and has a number of clinical and research interests, including hereditary cancer, pancreatic neoplasia, and laboratory administration. Cheryl is also a very nice and fun person, and I'm just reading from her bio that when she's not working as a pathologist, Dr. Matter is the ringleader of her own circus of 6 kids. So I would like to welcome Dr. Matter today to talk to us about cost-effective biomarker testing in lung cancer, the Alberta experiment. Thank you, Cheryl, for joining us today.
Thanks, Diana. Can everyone see my screen?
Yes.
Great. Okay. So we'll move on from here. So today I'm going to be discussing a proposed pathway for expanded testing and biomarker testing for non-small cell carcinoma, and in particular non-squamous non-small cell carcinoma for lung cancer patients throughout Alberta. Well, let's see if I can get it to move. There we go. Slow. So the objectives for today are to understand really what the international guidelines suggest for biomarker testing in lung adenocarcinoma, and what are the up and coming markers, because we know that the current biomarkers are fairly limited, but there are many, and every day more that I find out about, coming up in phase 2 and 3 trials. We'll talk about some of the challenges associated with stepwise testing in lung adenocarcinoma and analyze the pros and cons of using our proposed model, which is a tiered mass array followed by NGS model for testing lung cancer patients, and what the benefits might be to that and what their potential challenges are. I have no conflict of interest as far as I know that'll affect this presentation, although I do participate as a scientific advisor to a number of drug companies, particularly for the adoption of biomarkers across new indications. Moving directly into lung cancer in Alberta, we have about 950 patients a year provincially who are diagnosed with stage 4 non-squamous non-small cell adenocarcinoma. Basically adenocarcinoma or large cell neuroendocrine, and many of these patients are stage 3 or 4 at diagnosis. About half are stage 4. And we know that although survival is improving for these stage 4 patients, it was 17% in 1996 versus 28% in 2014, that's still pretty horrible. And most of those improvements are driven by targeted therapies, particularly for ALK and EGFR. The international guidelines that we use in Alberta for driving lung cancer biomarker testing are the IASLC, also CAP and AMP have come on board with this. So the International Association for Lung Cancer, the College of American Pathologists, and Association for Molecular Pathology, which says that all patients with lung adenocarcinoma should be tested for ALK, EGFR, and ROS1 mutations. The most recent guidelines actually state in black and white that multiplexed assays, particularly NGS, are preferred for the testing of these patients. And that if a multiplex test is adopted, that testing for BRAF, KRAS, HER2, MET, and RET is also highly encouraged. So there's just one problem. At the moment, and until the very near future here, we can't do that here. So what are we going to do? Now, the issue in Alberta is this. It's a publicly funded system, as is the rest of Canada, and complex biomarker testing hasn't been part of our paradigm until now. I will turn that down. Figuring out how to advance these to meet these guidelines while maintaining our current price points if we can, or minimally increasing our price points, has been kind of a challenging, ever-moving puzzle piece for us. So here's our first polling question, and Taylor, you're going to be taking over for the polling questions. But how many biomarkers does your lab currently test for in lung adenocarcinoma? So I think that's pretty good. So it's pretty well evenly split between 1 and 3 and 4 and 6, but nobody's doing, very few people, 17% of the population here, is testing for the recommended 7 or more markers that were in the international guidelines. And that's probably a reflection on the method of testing. So I'll show you what's been happening in Calgary and Edmonton, because until very recently we were separate organizations. And it wasn't until the late part of 2018 when Alberta Precision Laboratories, previously called Alberta Public Laboratories, was formed as a provincial entity. So right now, if you ask for this today in Calgary, you'll get a histologic evaluation of the lung cancer patient. And if it's adenocarcinoma or undifferentiated or mixed type, you'll end up sending a PDF to the laboratory, and your circle deepest H&E slide to the molecular clerk. You'll get a molecular panel that includes EGFR, and actually right now in Calgary, they're using a mass array panel that includes EGFR, KRAS, BRAF, and hotspots in HER2, as well as PIK3CA. And the ALK IHC is sent back to the original pathologist for interpretation. We also end up sending a PDL1 immunohistochemistry slide to Edmonton, which is evaluated by our IHC experts. If you're in Edmonton, it's fairly similar. You get your EGFR testing and your ALK testing and PDL1 testing. And as of July, you actually get reflexive ROS1 testing in both Calgary and Edmonton via IHC. So these are complicated pathways. They require multiple cut slides. And actually in Edmonton, it ends up taking 3 to 4 cuttings of a single block to get all of these slides sent to the appropriate stakeholders. So here's a question for the audience as well. Is the reflexive testing of lung adenocarcinoma available in your center? And looks like the vast majority of our participants say yes, reflexive testing is available. For those who said, what is reflexive testing? That's an important question and an important thing to define as a pathologist. Reflexive testing, as I'm defining it, is that a histology-based diagnosis, in this case lung adenocarcinoma, triggers a pathologist-ordered testing pathway. So for lung adenocarcinoma, we know these patients have relatively short survival timeframes. So any patient who is known to be stage 4 at diagnosis will get molecular testing ordered by the pathologist. And this is very important. The reason that it's important is because we want these patients to get timely and accurate test results that don't leave anybody out. The problem with our reflexive pathway is that, at least for us, we have to know it's stage 4. These patients don't get reflexive testing if they're stage 3 or below. And many times, if we're getting a lung biopsy, even with the imaging findings, we may not know if they're stage 3B, stage 3A, or stage 4 at the time of our histologic diagnosis. Now, here's an interesting look at lung adenocarcinoma testing in Calgary and Edmonton over years, just to show you that we've been getting better and better at identifying these patients. We know the lung cancer incidence in Alberta is actually stable or potentially even declining, but the number of biomarker tests ordered year over year has been increasing steadily. In Edmonton, this looks like a flat curve, but you can see it's only 2 years worth of data, which basically means that since we instituted the reflexive pathway, we've been keeping up with, or at least not declining in our ordering of this type of testing. The problem is this: the multiple different pathways for ordering lung cancer testing, EGFR by one platform in Edmonton, by one platform in Calgary, ALK IHC, ALK FISH, ROS IHC, and ROS FISH, is not cheap. In Alberta, the estimated cost for this testing is about $500,000 a year. And more significant is this. So these are representative images of EBUS biopsies of lung cancer. And what you can see is that these are little bitty pieces of tissue. Now, think about what I said earlier. 4 cuttings into a block to get every slide sent for EGFR, ALK, and ROS1. If you started with very little, by the time you get to EGFR, there's pretty much nothing left. So here's a question, and I don't expect you to know the answer, but it's just to stimulate discussion. I did this study from 2014 to 2018, and what proportion of our Edmonton lung biopsies had insufficient tissue for EGFR? The ideal question would be 0%, but I can tell you that's probably not correct. Okay, looks like we have a quorum. So the estimate here is about 15 to 19%. You're awfully close. Actually, ours was 12% for those that were performed in Edmonton, and 15% for those that were performed at our smaller centers in Red Deer and Grand Prairie. Part of the issue there is that the Edmonton guys use an 18 gauge needle because we have vascular surgeons on hand in case they hit something vital. The Red Deer and Grand Prairie guys use a 22 gauge needle because they don't want to poke a large hole in a very large artery. The bigger issue is this. If we get those small biopsies and we've cut all the way through them by the time we get to EGFR testing, EGFR is probably the largest hit biomarker other than KRAS, which is not currently actionable, in lung cancer. If we don't have enough tissue, those patients don't get tested. And in those 11% of cases here in Edmonton that didn't get tested, 75% of those patients never had a second biopsy. So we know that we're missing 2 to 3 patients a year at minimum who have EGFR mutations and would have benefited from that therapy. So the issue is this. If we lose tissue, patients don't get retested. And getting all the way through just the 4 biomarkers that we test currently happens only 90% of the time. The issue becomes there are 3, 4, 5 biomarkers in clinical trials, and we can't currently test any of those patients all the way through a pathway that would get them actionable results without losing tissue to the point of being more or less useless. Unless it's a large resection specimen, 75, 80% of our patients will run out of tissue by the 4th or 5th biomarker. And these are the biomarkers that we would like to test. So MET showing crizotinib responsiveness, RET fusions, and TRK. PIK3CA, there's some new drugs that are potentially useful for PIK3CA hotspot mutations. And KRAS G12C has 2 new drugs that are showing pretty strong clinical promise. Now, if we want to move to NGS-based testing for everybody, that's about $800 to $1,000 a patient. I can tell you right now that my executive leadership team won't pay for that. So we've proposed this for Alberta. We have a mass array platform which tests for hotspots in EGFR, HER2, KRAS, BRAF, and PIK3CA. And patients who are positive for these driver mutations, other than the PIK3CA, in general do not have associated mutations in ALK, RET, ROS, MET, or some of these other fusion drivers. So patients with positive testing by our mass array are proposed to not get further testing. And those who are negative, we'll move on to an NGS-based and RNA platform to look for fusions specifically in these driver genes. For those of you who are not familiar with the mass array technology, it's a fairly elegant way of looking at hotspot mutations based on MALDI-TOF mass spec. So MALDI-TOF is a very large mouthful that stands for matrix-associated laser desorption ionization, time of flight mass spectrometry. MALDI-TOF essentially uses endpoint PCR, similar to snapshot assays if you're familiar with those, where we have oligos built to the n-1 position of any hotspot, and we do a single base extension reaction. The results of that PCR are transferred onto a metal plate and covered with a matrix that facilitates the ionization of those molecules if they're hit by a laser. We can then hit that matrix and analyte with a laser and send it through an electric field generator or a time of flight tube to separate the fragments based on size and charge, the same way we would with any other mass spectrometer. The detector at the end can detect subtle differences in mass, so subtle that it can tell an A from a G. And this is the result. So in this case, we have a KRAS G12C mutation in our patient. So the G, which was the wild type allele, is replaced almost entirely by a T, which gives you the G12C mutation in that patient. It does that for multiple hotspots per well and multiple wells per patient so that we can get actionable hotspots in EGFR, HER2, KRAS, BRAF, and PIK3CA in the same assay. The benefits of this are that we have hotspot detection in multiple genes, and at least for many of these genes, the hotspots are all we really care about. The DNA input for a mass array is very low. It's only 10 nanograms, and it's very high sensitivity. So we did our limit of detection studies using a commercially available control that had T790M at 0.9%, an EGFR exon 19 mutation at 1.9%, and an EGFR L858R mutation at 2.9%. And we regularly detect, without having to do any fancy manual review, the L858R at 3%. However, we do see, more often than not, the EGFR mutation at 1.9%. And if we manually review the spectra, we almost always see the T790M at 0.9%. We've also seen, and much to our chagrin, that we have better detection of the EGFR exon 20 insertion mutations than our previous QIAGEN kit was able to detect. This has become something of a problem for our clinical trials because they require an FDA-approved assay to detect the EGFR exon 20 mutations. So when we detect it by mass array and we go back and try to detect it with our EGFR QIAGEN kit, we can't. And these patients are no longer eligible for the clinical trial despite the fact that we're reasonably confident that they have an eligible mutation. Mass array is also dirt cheap. With tech time and reagents, it's $236 a sample, which those of you who do molecular testing know is a bargain, especially when you're getting multiple hotspots for the same assay. Our proposed algorithm in Alberta takes the mass array and then splits it into either an Edmonton-based Oncomine Focus assay with an RNA input or a Calgary-based Archer RNA lung fusionplex that has slightly different costs and are negotiated differently through vendors. But the difference compared to our current assay is only about $10,000. This is really important because not only are we covering the 4 analytes that we would currently cover, and we will still continue to cover PDL1 by IHC because there's no molecular equivalent, but we're covering all of the up-and-coming biomarkers that are available in clinical trials. The downsides of mass array are that it's limited to those hotspot mutations. The hands-on time for our technologists is moderately high. There's 2 PCR steps, a cleanup step, and then you have to load it onto the machine. So it takes a full day. And there's no detection of fusion genes, although a fusion panel is available. We know that it's less sensitive than our NGS-based panels. So for our fusion genes and our MET exon 14 skipping, we have 2 potential NGS panels that'll be used in Edmonton and Calgary respectively. And they cover essentially the same targets. The question is, why be different north and south? So APL is a provincial entity, and we have committed to providing equivalent care for all patients across Alberta. But there are benefits to having some differences across sites. So for example, Edmonton has the Ion Torrent S5 and the Ion Chef platform, which allows us to do automated library preparation and sequencing for 8 patients at a time. And it works with the commercially available Oncomine Focus RNA panel, which we've already spent money validating. And that became kind of a big issue when we brought it to our clinical and executive leadership teams. In the South, we just acquired a Genexus system, which is also a thermo-based chemistry, but combines the library preparation and sequencing into a single instrument and has a little bit more flexibility. Our molecular pathologists in the South elected to use the Lung Archer Fusionplex, or the Archer Lung Fusionplex panel, which offers similar capabilities, but slightly higher sensitivity than the Oncomine Focus panel. And because they're similar chemistries, we can send cases down to Calgary if we have unexpected downtimes, or they can send cases to us. We also have the capability of using the raw output files in bioinformatics to share across sites so that if something is sent from Calgary to Edmonton, the guys in Calgary can sign it out. Or if it's sent from us to them, we can get the bioinformatic pipeline output and actually report it ourselves. But the point is that the different but equivalent assays will allow us to orthogonally verify unusual or unexpected variants, and if necessary, we can focus on the things that the panels do differently for those rare patients who need the full shebang type testing. The benefits of this algorithm, at least as we've proposed it to our executive leadership team, is that it covers all clinically actionable targets, and most of the targets being covered in clinical trials. We admit, and we'll say with no chagrin, that we're never going to cover it all. Most of these trials are changing daily, weekly, and while we may try to future-proof by creating the most complex panel we can for the lowest cost that we can figure out, we are always going to be cognizant that new markers are going to be coming, and we will have to reevaluate these panels as those markers evolve. The benefit of doing it this way is that we have the capability of doing a single DNA and RNA extraction for all patients at the time of receipt, which means that we only have to extract it once. That's one cutting, not four. Well, actually two cuttings, because we still have the IHC cutting for PD-L1. And we anticipate that it should decrease the number of patients who have inadequate tissue from 10% to less than 2%. The screening with mass array is substantially cheaper than the NGS. When we looked at doing this, every patient with NGS, it was going to cost on the order of 10x compared to what we were proposing over the current increased cost. So about $100,000 a year instead of $10,000 a year cost to the system. And so it is the belief of both the North and South molecular pathology groups that by offering this proposed tiered panel, that we'll be offering the most comprehensive testing for the lowest cost. So, my question to people watching today is, would you consider using such an algorithm or something similar in your centers for the detection of lung biomarkers? And we'll discuss hopefully in the question and answer period, what you're doing in your centers, what the considerations are, and where you're thinking about moving. So just to conclude, before I open this up for questions and discussion, the idea behind the proposed algorithm in Alberta was a recognized clinical need and need to meet up with internationally respected guidelines. It's our belief, although we are still evaluating other pathways, that the best and most cost-effective way to do this would be to combine our mass array and NGS capabilities to standardize North and South and make these biomarkers available to all Albertans. There are initial and ongoing costs associated with this paradigm. And I haven't discussed this in any detail, but we know that at least for some of our biomarkers, the drug funding is being tied to the biomarker testing funding. So we are starting to evaluate new paradigms for covering these types of mutational assays and what that'll look like going forward for our patients in this publicly funded system. So at this point, I'll open it up for questions and discussion. I'm hoping that our panelists or our audience members here will be willing to share their own experiences with lung biomarker testing and other, multiple biomarker testing across histologies to figure out where people are right now, where they're going, and what the considerations are across the world for expanding such testing, given the relatively limited resources that we all face.
We do have quite a bit of time for questions and for discussions. And as we're waiting for the questions through the chat, I would like to ask you the first question maybe. When you validated your new technologies, how did you deal with the samples that you need for the fusions with low incidence, for example, NTRK? And when you did you validate, for example, for all the 3 NTRKs, or how did it work? What number of samples?
I think that's a major challenge actually across all multiplexed assays. is we know that we can't validate every potential variant that we will ever see. So for us, at least initially, we know that we will do our validations with what we can find. For NTRK, for example, we have a handful of samples with HEV6 and TRK3. We probably won't find in our validation set any NTRK1 or 2 samples. What that will necessitate as we go forward is that any patient who's flagged as positive on our assay with these rare fusions will have to be orthogonally confirmed, either by sending to Calgary for their Archer technology or to do IHC for NTRK, for example, to make sure that it's positive where we would expect it to be positive. We know that we can't just expect it to work perfectly, and that anything that we identify that is outside of our validation set will have to be orthogonally confirmed, at least for the first few incidences.
Okay. How do you deal with the quality assurance for this test?
So for us, we tend to use multiplexed controls that we have known variant allele fractions for. So for example, for lung fusions, we have some commercially available controls that have ALK, RET, and ROS fusions available. And we tend to either dilute them down to our limit of detection to make sure that we're performing up to spec, or we will buy controls that have a variable range of variant allele fractions so that we can figure out whether we're detecting things that we expect to detect. We have some fairly strict quality measures in place for things like read depth and for expected variance. We try to make sure that every sample on the run meets those quality thresholds. And we also participate in both commercial, if available, and inter-laboratory proficiency testing wherever possible, so that we know that we're getting the same results as our colleagues across the world.
Thank you. I apologize if I missed this in your presentation, but what's the turnaround time for your testing? And maybe if you don't mind to define the turnaround time, just to make sure we're talking about
Sure. So turnaround time for us is from the receipt in lab to the publication of the patient report in the electronic medical record. You could define turnaround time as from time of order to time of report, but what we find is actually that the time of order to time of receipt is highly variable depending on the person ordering. So what we would like to have, and we are anticipating, is less than 10 business days from time of receipt in lab to time of reporting. What we anticipate will probably be more realistic is for ALK EGFR, or for EGFR and KRAS, BRAF, the things that are available in the mass array, we can probably turn those around in less than 7 business days. But those that are going to require NGS testing, which we anticipate will be about half the cases, will probably end up being a 3-week turnaround time, just because we can only run our panel once a week in order to fill batches.
Okay, and if I can follow up with a question, if the turnaround for the fusions it's about 3 weeks, then wasn't it difficult to give up ALK and ROS IHC? Were the oncologists happy with that instead of having a couple of days turnaround time for the ALK and ROS? Also, the fact that it influences immunotherapy as well.
Right.
The oncologist cannot treat with immunotherapy unless the patient is proven not to have at least EGFR and ALK mutations.
So certainly those are things that we're discussing with our oncology colleagues. For ALK, a positive result is considered equivalent to a positive FISH result, but for ROS1 it's not. And so for ROS1, positive or equivocal results will end up needing FISH testing anyway, which right now we're sending out, and that can take 2 to 3 weeks to turn around. For ALK, equivalent or equivocal results by IHC require FISH testing, which takes us an extra week to turn around as well. So the overall turnaround time for at least equivocal testing is not that different. And the hope is that a large proportion of these patients won't need any kind of fusion testing at all, and will have their EGFR, KRAS, or BRAF results that would drive testing or drive clinical therapy within that 7-day period. Now, the issue is, as you say, you can't treat without at least proving the negative, which can be a problem in these patients. So the goal actually is to increase our reflexive testing, because our biggest delay in turnaround time is not actually in the lab. The biggest delay is actually getting the oncologist to see the patient, order the test, and get it sent to us. So I think that, overall for clinical care, what we're going to find is our outreach to our clinical teams and our increasing of reflexive testing, potentially to all patients for EGFR, as there's data now in stage 1 and 2 patients for osimertinib therapy in EGFR mutated lung cancer, may actually improve our turnaround times just by decreasing that first and most onerous step of getting the oncologist to order.
Okay. Yeah, it looks like that's it's almost a national revolution that we're trying to do in lung cancer in Canada to get into reflex testing, to allow pathologists to order molecular testing at the time of diagnosis. And as you mentioned before, everything it's really just funding, because at the time of diagnosis, often pathologists are not aware of the stage. So we will probably over-test a little bit stage 1, 2, and 3A, but I guess that's still better given the fact that a large number of lung cancer recur, and that information will be useful for the oncologist at the time when the patient will become advanced stage. Maybe I will take one of the questions from the audience, kind of to wrap up our discussion here. If you were to decide on an ideal test for ALK, do you consider that IHC or new generation sequencing is the ideal test?
I think it depends a bit on your next generation sequencing test. So IHC for ALK is highly sensitive, but we know that it's slightly under specific. You can get a positive result without a fusion. The only question is, do those patients respond to crizotinib without fusions and just overexpression? Ideally, we would like NGS just because it does decrease the number of cuttings on the tissue. Now, we would like our NGS to be as sensitive as the IHC. And certainly panels that are designed with specific partners on both sides are less sensitive, although more specific, than the IHC markers. Whereas panels that are such as Archer, which have the capability of detecting unknown fusion partners, are probably just as sensitive. For most of the patients that we see, the likelihood of an ALK fusion pretest probability is quite low, so we would consider them fairly equivalent. And given that the NGS panels will detect not just ALK fusions, but RET, ROS, and MET exon 14 skipping, I think that's probably the way that most labs, if they have the funds and technology available, will end up going.
Sure. If your budget would be unlimited, what would be your ideal workflow for, I assume, for non-small cell lung cancer?
Well, I think if our budget was unlimited, we would all probably prefer to do reflexive testing of all lung adenocarcinoma patients at the time of diagnosis using an NGS-based DNA and RNA panel. The reason for that being that it would be a single workflow for the laboratory, which would be ideal for us, would allow us to make sure no patient falls through the cracks with minimal wasting of tissue, and would give us the most rapid turnaround time available. That said, budgetary limitations being what they are, I think we're all kind of searching around in the dark for the best possible solution that costs us the least that we can afford.
Yes, and I guess in lung also the availability of the tissue, it's another big issue. I didn't mean to rhyme, but I did. So another question. Is anyone aware of multiplex IHC in a clinical space that could be equivalent to the mass array platform you presented?
I haven't heard about that, but I will easily admit that IHC is not my area of expertise. I'd be very interested to hear about it if is a multiplexed or small panel of IHC that could detect a fair number of these alterations.
No, I'm not aware of any myself, but if anybody in the audience has a comment, please just put it in a chat and I'd be happy to bring it to everybody's attention. Another question that we have is, against what method have you validated the mass array method?
So we've validated our mass array against multiple methods. For EGFR mutations, we validated it against our QIAGEN kit, which we'd used for many years for the detection of EGFR mutations. For our PIK3CA mutations, we actually validated it against additional testing that had been done in a clinical research sphere to detect PIK3CA mutations. And for our KRAS mutations, we validated it against our snapshot assay, which we had previously used for formalin-fixed paraffin-embedded tissue predominantly in colorectal and melanoma, but had also used in other FFPE-based diagnostic pathways.
Okay, we got a comment that IHC cannot accurately detect this type of molecular alterations. I don't know if you want to comment on that. I agree with this as a general statement. What I would like, maybe more for the pathologist in the audience than for medical oncologists, as pathologists, we don't really often read a drug label. For me, since all my involvement in precision medicine, I learned to go backwards. I start with the drug label. And depending on the approval in your own country, you would learn the limitations or the freedom you have in the laboratories to implement certain tests. And if I can historically give the example of ALK, the crizotinib FDA approval initially was based on a companion diagnostic test, which locked the testing to the FISH probe only. While not too many months later, Health Canada approval for crizotinib allowed the laboratories to use any, and quotes, valid test. So in Canada, we implemented IHC screening followed by FISH and later on NGS confirmation of equivocal cases, while US was stuck for a long time on having to perform FISH because that was the companion diagnostic test required in the drug label. So I agree that from a scientific point of view, IHC cannot accurately detect this type of molecular alterations. However, the crizotinib's drug label again in US have changed, and the companion diagnostic test that it's required right now, it's IHC. A certain IHC test, but again, if you go backwards from the drug label, it's going to teach you a little bit of what you can use in the laboratory for testing for different rearrangements of or mutations. So I don't know, Cheryl, if you have anything to say about this.
I would say that that's a fairly common problem, especially with FDA approvals. We know for BRAF, for example, the original FDA approval tied it to the Roche diagnostic test, which we knew didn't detect some of the non-V600E mutations. In Canada, at least historically, the Health Canada approvals have been more open in their language and interpretation of valid tests for the detection of biomarkers. And I don't think we will see any new test or new drug come on that would require a very specific diagnostic pathway. They typically will accept that the laboratories know what they're doing and that LDTs are an acceptable pathway for biomarker testing. But I do know that it is a problem in the US and can be a problem elsewhere in the world, and definitely ties your hands a bit when it comes to trying to create alternative pathways that might be better bang for your buck, for example, in terms of getting what patients need for the lowest cost.
Okay, thank you. We have two more questions. One is there any thought of the ctDNA test at diagnosis in non-small cell lung cancer in Alberta? And then I'll bring up the next question after you answer this one.
So for those patients who are not amenable to biopsy, we do have available an EGFR cell-free DNA test run in Calgary. Classically, we have not offered cell-free DNA testing as primary diagnosis unless the patients either don't have tissue available or are too frail to get a second biopsy. The reason for that being that classically, cell-free DNA testing has lower sensitivity than tissue-based testing for the detection of molecular alterations. Now, as cell-free DNA testing gets better and less expensive, that may change in our patient populations, particularly as we've said, half of patients with lung cancer are stage 4 at the time of diagnosis. And a fair proportion of them may be too frail for significant tissue testing. And in those patients, having a cell-free DNA option would be better than nothing for sure, and would allow them some options that may not otherwise be available. That said, cell-free DNA testing, at least at the moment, is more expensive than tissue-based testing just because of the depth of coverage needed to detect alterations with certainty.
Talking about the cost of the test, in the setting of resistance, when you do cell-free DNA testing in Alberta, what is the funding? 1 or 2 tests followed by tissue, 1 test followed by tissue if negative? What's the algorithm that you are funded for?
So in general, things just get passed through without too much evaluation of where the money's coming from, which is kind of a problem. Classically, our patients will get 1 cell-free DNA test followed by a tissue biopsy. In those patients that have EGFR mutations where you're looking for resistance, you can at least detect the original driver mutation in the cell-free DNA test to know whether you're sufficiently sensitive to detect a resistance mutation that's occurring at frequencies that you would expect to be clinically actionable. So if the patients don't detect their EGFR mutation in the cell-free DNA assay, we move directly to tissue testing because we know that we were completely insensitive to finding their mutation.
Okay. Another question is there a difference in the turnaround time from rural versus Edmonton/Calgary area for testing? And do you, I guess that's a separate question. Do you have any thoughts on a point of care solution? Maybe
so we can split these questions into two. Certainly there is a difference in turnaround times for rural versus urban based testing. And the big difference there is the time from order to receipt. Our rural-based patients have multiple cards stacked against them in that respect. One, they have to transport the tissue or the cell blocks or pull the cell blocks from Dynalife, which is based in Edmonton for their North Sector patients. But the bigger issue there actually for our rural patients is access to oncologists and access to ordering. We know that many of our patients don't see oncologists in the same time frame, or if they do, they may not always be upfront with testing and ordering, which is a problem for our rural patients. Fortunately, at least in Edmonton, all of our rural tissue biopsies come through the Dynalife site here in Edmonton. So our tissue blocks are actually in the city at the time of ordering. So once the oncologist orders, and once Epic goes live across the province, this will be even more streamlined. Once the order is put in, it's not that we have to bring the tissue from Grand Prairie to Edmonton. We just have to pull it from Dynalife and send it to the U. So that's, shortens our turnaround time a bit.
Yeah.
But overall, yes, I think our rural patients are at a significant disadvantage to our urban ones when it comes to testing. not least because our oncologists here might know of clinical testing programs that are not available if you're, you know, not deeply enmeshed in the clinical trials program here at the urban sites. In terms of point of care testing, that is a very complex metric. We have a lot of possible solutions that would be definitely better than not having testing at all, but they have associated costs and they don't offer the full comprehensive
Answer.
testing that we would prefer to get for all of our patients. And there's definitely economies of scale that are achieved by having batch testing. And if we split out into point of care across our rural sites, it does decrease our efficiencies here in the urban sites for testing as well. So there's a very complex dance between, you know, ideal patient care, turnaround time, cost, and efficiencies of scale that have to be considered when you take on a point of care test for the purposes of improving that rural turnaround time.
Yes. You presented us the Alberta experience, and that's the experience from the point of view of a referral molecular lab, you know, covering, um, I would say, half of the Alberta uh, patients, testing for half of the patients in Alberta. I think on November 5th, on the next Camppods, we will hear from Dr. Brandon Sheffield, and he will share with us his experience in a smaller molecular lab and how he successfully managed to decrease the turnaround time, implement something that we call point of care molecular testing. And I think I would like to maybe end with another question. Do you have feedback from oncologists about the clinical response in patient positive for the biomarker tested? The reason I wanted to kind of end with this is as Dr. Miloski and I presented on the first Camppods, if you have a strong relationship with the oncologist in your center, their patients become your patients. And although we don't see them as pathologists, I am often extremely curious what happens to this patient. And sometimes the oncologists do volunteer to give us feedback on what happens to these patients, but it's not rarely that I actually pick up the phone or send an email. Hey, what happened with our young patient? Or, you know, whatever, we have a kind of a nickname for these cases that we share. I think that's also what happens in a smaller hospital, and that's what we're gonna hear on the November 5th presentation, because it really is the partnership between pathology and oncology that has changed the molecular testing landscape in Brampton, Ontario, where Dr. Sheffield works. And I don't know what your interaction as a molecular pathologist in a referral lab is with the oncologists, given that they are geographically remote.
So for us in Edmonton, our interactions with the oncologists have historically been fairly uh, distant, I would say. You know, they would contact us if they had a problem or wanted to complain, but otherwise we didn't hear a whole lot. Um, that has changed in the last couple of years as we've done more outreach as a group to, you know, bring our oncology colleagues into the discussion about where we are and where we're going. And now I get regular correspondence from our oncology colleagues about, you know, can we test for this? What about that? How do we go about evaluating this rare or unexpected finding? And it really does improve our ability to figure out what the up and coming things are, what our oncologists are interested in, what our patients need, as well as help us close the loop in terms of these relatively unexpected or unusual things that otherwise just, you know, go into the bin of the interesting things that I hope to see again in my career. So I think, you know, certainly oncology outreach is important, whether you're in a small center where you might happen to know that the patients are your neighbors, or whether you're in a large center where your patients are, you know, half of the province. but it does take a little more effort here, I think, and a little more outreach on our part to figure out where that starts.
Okay. So I would like to thank you very much for your presentation and for the discussion. I hope the audience found it interesting and got some practical points that they can apply in their own practices as they return to their hospitals. I would also like to thank the audience for all the questions that they send. It looks like there was a lot of interest in the subject, and also if you have any comments, any ideas for future Camppods, please fill up the evaluations, which will be coming, will be sent to you by email. It is very important for us, not only for accreditation purposes, but also for me personally as a chair of this activity. I read all these comments with high interest, and I learn a lot from those comments, and I try to apply them on future activities. So please do fill up the evaluations and give us your feedback, good or bad. So you can leave it in here, because I can end the activity by thanking again our sponsors, AstraZeneca, Bayer, and Merck, for their educational grants in support of the Camppods. And I would like to invite you for November 5th. Maybe if I can have that slide. If I can make a suggestion, please, when you do register for the seminar, also register on Zoom, because otherwise you won't be able to join. We apologize for the double registration, but it looks like that's the requirement for being able to participate. And here we are on Thursday, November 5th, point of care molecular testing with Dr. Brandon Sheffield. We will learn to gain an appreciation for the urgency with which biomarker testing needs to be completed, to identify ways that surgical pathology practice can facilitate cancer care in the clinic, and build familiarity with emerging techniques in molecular pathology. And with this being said, I wish you all a great day in North America, and good night in other parts of the world, and stay safe, and we'll see you again on November 5th. Thank you.